30s. There you are. I'm back again for another lesson. We are going to be starting a new unit today. We're going to be talking about population genetics. Specifically, we're going to focus on the Hardy-Weinberg equation, as well as how populations can change over time in terms of their genes. So just a little bit of introductory vocabulary for you. Population genetics is simply the study of the genetic composition of, you guessed it, a population. So previously we had looked at individual genetics. So this individual has brown eyes, this individual has blue eyes. Now we're looking at, okay, how many people in the population has blue eyes, how many people in the population have blue eyes, and how many people in the population have brown eyes. Population, group of interbreeding individuals, same species in a particular area at a given time. And a gene pool is a collection of all the alleles and their frequencies within this population. So you can think of all of the genes possible in that species would be within the gene pool. So what is the Hardy-Weinberg equation? Well, when we look at a gene pool, we can start to get gene frequencies. So for example, what's the most common blood type here in North America? It's actually blood type O, whereas blood type AB is considered quite rare. So even though we have different alleles or different genes in our gene pool, some tend to show up more frequently than others, and this is why we study population genetics. Another example is the recessive Rh allele. Only about 15% of Canadians are Rh negative. Then we can look at a bunch of frequencies here. So we can have our genotype frequency. So this would be the genotype of how many individuals are homozygous dominant, how many individuals are heterozygous, and how many individuals are homozygous recessive. We can then do phenotype frequencies, so how many people have the dominant phenotype, how many people have the recessive phenotype, and we can even do allele frequencies, so how many people contain the dominant allele and how many people have the recessive allele. So I'll show you what this looks like here. So let's take this mouse example. We can see that black being the dominant trait, white being the recessive trait. We can see our three genotypes, right? So then out of 200 mice, if 72 of them were if 72 of them were homozygous dominant, that means 36% of the population is homozygous dominant. If 96 of them were heterozygous, that means 48% of the population would be heterozygous. And if 32 of them were homozygous recessive, 16% of the population would be recessive. Then if we look at our allele frequency, we can notice that, okay, if you're homozygous dominant, you have two copies of the big B allele, so 72 times 2, that means there's a total of 144 big B alleles. Also, we need to factor in the 96 that are part of our hybrids, because when we look at a hybrid, one allele is dominant and one allele is recessive. So this gives a 60% allele frequency of the dominant allele, not the dominant genotype, not the dominant phenotype, just the big B allele. Then we go over here, factoring in the hybrids with the recessive allele, and we get 40%. And look at that, 40 plus 60, you guessed it, adds up to 100%. We could also do a phenotype frequency. So what we would do is we would say, okay, 72 plus 96, that would be our dominant phenotype, and then 32 would be our recessive phenotype. So that's kind of basics on how we calculate these frequencies. Now, there's a mathematical way to do this. It's called the Hardy-Weinberg equation. Essentially, to do this, you would need to make sure that your population is what's called genetic equilibrium. Your population needs to be in genetic equilibrium for this equation to function properly. So essentially, you need to have a large population, because small populations can leave things up to chance, right? If there's 10 individuals, an earthquake comes along, it takes out 5, that's half the population. If there's 1 million individuals and 5 are taken out by the earthquake, then that doesn't make as big of a dent. Random mating, so you can't have any sexual selection whatsoever, can't have any mutations, no migration, so individuals can't leave the population or enter the population, and there needs to be equal viability, fertility, and mating ability, meaning there can't be any natural selection. Now, can you think of a population in Hardy-Weinberg right now? You probably can't, because when we actually take this and apply it to the real world, there's no population out there where there's no natural selection or no mutations or random mating, right? They might fit some pieces of Hardy-Weinberg, but not all. So why study Hardy-Weinberg? Well, it will give us a good idea or a general idea of where the population is at. But we need to remember that populations will change over time. This is how evolution works, right? The gene pool and the population will be undergoing changes over time. So when we look at the Hardy-Weinberg equation, a few things to look at here. Number one, we have p squared plus 2pq plus q squared equals 1. So this will be the equation that we use to solve for our various components related to our Hardy-Weinberg equation. So let's look at p squared first. P squared is taking the frequency of the homozygous dominant individual. So when you solve for p squared, you're solving for big B, big B. 2pq solves for the frequency of the heterozygous or carriers, so B or big B, little b. Q squared is the frequency of the homozygous recessive, so that would be the little b, little b. Finally, if you want to solve for the allele frequencies, we have P and Q, where P is representative of the dominant allele, Q is representative of the recessive allele. So essentially we can have another equation here where P plus Q equals 1. And this is important because this equation is where we get this equation from. Because if we have two alleles, right, because we know we have two alleles, what we can do is essentially FOIL, for you math people out there, this equation. All right, what does this give us? First, P times P, P squared, PQ, PQ, so that's 2 PQ, and then finally our last Q squared. So that's where this equation comes from. Okay, so let's put it into action here. Let's do a little practice problem. So when we're doing these Hardy-Weinberg practice problems, a few steps to consider. Number one, and I think step number one is very important, write down what you know and what you don't know so you can kind of map out where you need to go and what you're trying to figure out. So remembering what all of these things mean, being like, okay, I know P, I might know Q, or I don't know Q squared, and you're trying to figure out what your unknowns are. Step 2, solve for your unknowns. Step 3, check your answer. Great thing about Hardy-Weinberg is you can always check your answer because everything should add up to 1. So let's go through a little example here. Let's say 15% of squirrels have gray fur, which is the recessive phenotype. What are the allele and genotype frequencies? So we know that little b, which relates to Q squared, is 15%, which is going to equal 0.15. All right, our first step will be to do this. We can't solve for P, can't solve for P squared because we don't know it. We need to solve for Q first because this is the only variable we can solve for. Now, how will we solve for Q? Well, if it's squared, the way around that is to square root both sides. So we're going to square root both sides, so our Q will equal 0.39. So this means 39% of the population has at least one, right, little b allele. Then to solve for P, we're going to take 1 minus Q, because this will give us our P, which is going to be 0.61, okay? So now that we have Q squared, P and Q, we can solve for the other unknowns. So moving down here, Q squared we already know to be 0.15 because they gave it to us. P squared, we're simply going to take 0.61. We're going to square that, which will give us our P squared, which is going to be 0.38. And finally, 2PQ, we're going to take 2 multiplied by P multiplied by Q, which will give us our heterozygous frequency, which is going to be 0.47. Then, if you were to add all three of these together, you need to make sure they add up to 100% or 1, which they do, and you have yourself a successful Hardy-Weinberg equation. Now, moving on here, as I mentioned, most populations, or all populations, won't be in Hardy-Weinberg because evolution. Changes in the gene pool over time. That is essentially evolution. So how can the gene pool change? Mutations can change it. Gene flow, non-random mating, some type of selection, as well as genetic drift. All of these factors can change the gene pool over time, leading to evolution. Now, some factors will increase your genetic variation, other factors may decrease your genetic variation. So for example, a mutation will lead to increased genetic variation, but these mutations could be good, right? They can give you an advantage. They could be bad, they could be lethal, so it's a disadvantage, or they might be neutral, no advantage or disadvantage. However, a mutation will always add in a new allele or a new variation. Recombination, so crossing over. This too will increase your genetic variation because now we're shuffling the genes, we're going through the process of crossing over, giving us new gene combinations and increasing the variation within the population. Gene flow. Gene flow is essentially migration where individuals are leaving a population and entering into a new population, so essentially immigration. When the individuals leave the population, they take their genes with it and move it to the new population. So now this population here that they're immigrating to is gaining alleles or gaining variation, and now this population has lost variation. So when we think of immigration, that's a way to increase variation, and essentially emigration will be a way to decrease variation. Now, other ways that we can decrease variation, of course, natural selection. Natural selection will be essentially the environment or the ecosystem is going to select which traits are most desirable. Those traits will allow that organism to have a selective advantage, pass those traits on to their offspring, and away we go. Organisms that don't have this selective advantage or have unfit genotypes tend to be removed from the gene pool. So now we're kind of whittling down our variation to the most desirable version. Sexual selection, another example here. Essentially, we're going to have individuals, sometimes males, sometimes females, depending on the species, which will choose their partner for a variety of reasons, right? The brightest colored peacock, the moose with the biggest antlers, or who wins the battle between the males. It's usually in nature the males that are doing some type of courtship, whether it be a dance, whether it be brightly colored feathers, whether it be a battle of some kind, and then it's the females that are choosing, right? They're going to choose which male is the most desirable based on certain characteristics. Also, genetic drift. Genetic drift will also decrease our genetic variation. So it's essentially random changes which leads to a change in the allele frequencies. We have red frogs, green frogs, and yellow frogs. If all the red frogs die out, and here's our new population, look how our allele frequency has changed, right? We have now changed what frogs we can have in that population. And it generally has the greatest impact when the population is small. Now, the founder effect is another example here where individuals from a larger population will leave to establish a new population. When they do that, the allele frequencies will be different because if a small group leaves the large group, whatever alleles are in that small group is kind of your baseline for your new population. So for example, the Amish people in Pennsylvania are descendants of people who emigrated from Sweden in 1720. And those people that entered into Pennsylvania and brought along their traits started essentially the gene pool, which would be different from the gene pool back in Sweden. Another example here that we can show is the bottleneck effect. So what happens with the bottleneck effect is there's a severe event of some kind, a natural disaster, sometimes humans overhunting, and the surviving population now makes up the gene pool. So the surviving population might have a gene frequency or allele frequency completely different than the original population. Example here is the northern elephant seal. Basically, they were overhunted by humans. So we have our original population here. Let's say these are the surviving individuals, and then they're able to repopulate. That next generation looks completely different than the parent generation. Now, thankfully, the elephant seals have begun to rebound, but their gene pool or their allele frequencies will be different than before they were overhunted. So that's all I have for you today in terms of gene frequencies, increasing genetic variation, decreasing genetic variation, and of course the Hardy-Weinberg equation. Next lesson, we're going to talk about population changes, so how population numbers can increase and decrease, showing you different types of growth rates, as well as population density equations. So, thanks again for watching, and we'll chat soon.